right, thank you very much for having me back. Lucky me, I got 2 slots. So this is the second project that I'm involved in, and unlike the other one that was kind of new project, this one I've been running for the last 2.5 years. It's to make eDNA data fair. And Christian will be proud of myself. Since I started this project, I stopped writing papers, and then I've been working on this. Yeah. So FAIR stands for Findable, Accessible, Interoperable, and Reusable. In this talk, I would like to introduce about what is FAIR principles, and then why do we need to make eDNA data fair, how are we going to do this, and then what kind of activities that we've been running. So first of all, what is FAIR principles? Open data, I'm sure many of you are very familiar with this term. Open data is about transparency and reproducibility of data, of a study itself, and then also data. But FAIR data goes beyond that, and it's about reusing the data in order to generate a further knowledge at speed. So it's a cost-saving measure, like the data is already there, we are recycling, reusing it. And also it's about long-term value of data. Why do we need to make eDNA data fair? It's because we need a better, more robust environmental monitoring, the biodiversity monitoring tools, because of number of environmental effects around the world. And because I think we all agree that eDNA is a very robust tool for that. And because of the robustness of eDNA tool, number of eDNA publication is increasing. As you can see on these 3 graphs, they're targeting a different ecosystems, freshwater, aquatic, or terrestrial, but the patterns are all the same. Number of publications increasing very rapidly in the last 10 years or so. And each of these eDNA papers produce tremendous amount of data. FASTQ files, the DNA sequences, species occurrence data, and all of those are really valuable for environmental conservation. And this is all about publications, but there are so many other initiatives like ANEMONE database and many big scale projects that are producing a lot of data. So I would like to also highlight that part. And if we have all this data in a fair manner, what can we do with it? What kind of benefits are out there? This is one of the benefits for data providers, that if you make your data open and fair, it's been proven by multiple studies that your studies will become more visible so that you can get more citations. And that's one of the great advantage for researchers, I think. And for data reusing side, there are so many benefits. We can run further studies at extended time and space that is impossible to do within a single project. We can increase species identification accuracy because DNA reference library are improving rapidly as well. So we can reanalyze those existing sequences, eDNA sequences, and then improve that part. Invasive species frontline monitoring is another opportunity that we have. If we have a big database from eDNA, then we can use in this space. And through reusing the data from different labs, regions, countries, we can enhance collaboration. And then through all these activities, we can generate knowledge at speed, and ultimately we can enhance the data-driven decision-making. Hopefully. So at this moment, what is this eDNA data life cycle like? Are they open? Are they fair? The answer is they are open, but unfortunately not fair yet. The 2 major reasons. One is that the data is not standardized. So data might be open through Dryad, Zenodo, those open data repositories, but the format is not standardized. So as many of you mentioned in the last few talks, if it's not standardized, then we cannot really put the data together and then make the comparative studies. Second reason is incomplete data, like this journal article says. Some data are archived, but they're half empty. Some important information that data reusers want to look at, they're missing in the metadata. But it's changing, and I'm really hopeful that it's going to change even further. So there are so many projects now out there, regional level, project-based database are there. ANEMONE database is a great example. And then the other institutions are also having their data, lots of samples, and then everything is publicly open. And some of them would also go beyond this project-based database, but they will publish the data to international databases. So through those databases, multiple projects' data can be together, and then data reuser side, we can just go to one resource and then get the data and reuse them. But there are multiple bottlenecks, challenges to make data fair, and obvious reason is the technical and the resource limitations. Everyone is busy, data formatting and the publication, it takes time, so that's one bottleneck. And then some people might be willing to do that, but then they might not have the skills or tools. They don't know where to find the standard guidelines and things like that. Maybe the standard guidelines are still missing as well. And that's one of the reasons why it's still challenging. Other type of barriers is the perceived barriers, and lots of like this kind of paper, more like psychological side of researchers, they identify multiple perceived barriers. And one is that people might not be aware about fair data values, or they might be worried that if I make a data open and then people might find mistakes in the data. And resistance to openly sharing data, I think this is the major part. And every time when I give a talk about fair principles stuff, then I get a comment about this, and I completely agree. Everyone spends so much time and effort to find the money to go and collect the data, process the samples, analyze, and then finally have a data, and then make that completely free for anyone to just take and use. You might feel a bit hesitant about it. So I believe like if we all agree with the values of fair data, then maybe we can overcome these kind of things. But then there are also a lot of communications, discussions are required, I believe, and hopefully we can overcome those things. And to overcome that, we started this project. So this is the FAIR eDNA project. We make abbreviation, we call it FAIRY project. And this is the infographic of the project. So starting from eDNA processes, maybe I walk around and point out. So here people start with the processing, and then they generate the data, identify the species. At this moment, the data is not standardized, so these pieces of puzzle from different studies, they don't fit together. But through these activities, we want to change the life cycle of this data. And then ultimately, the outcome is that those pieces of puzzle will fit together, not only about eDNA data set, but other type of biodiversity data from different methods. They can plug in together, and then we can do all sort of amazing things. So I'm going to walk you through a little bit about this middle part, what we've been doing. So this is what we are working a lot through this project. And you can see that, so we published this guideline last year. You can see a lot of co-authors, so it's an international collaboration, and then it's really a community-driven initiative. I will direct you to the website rather than this paper, to be honest, because this is all the paper contents are included in this website, as well as more updated information. So after we publish, we continue working a lot to keep on improving, adding on extra tools and stuff like that. So everything will be on that website. So the first thing we did at the start of the project is to establish the working group. And we started very small, but as soon as I started communicating with multiple people, it got big very quickly. So we are 32 members. I think we actually have a few more now, and 25 institutions. And this growth of this working group really reflects the needs that everyone was recognizing already about this fair eDNA data. We needed to do something about it. This group really consists of multidisciplinary teams. One type of organization is a database. And then we also have a data standard, so biodiversity information standard and genetics standard. Then we also have a journal editors as well, and I'm going to touch a bit about this, why we have this journal team a bit later on. with this working group, what we did is to review existing data standards. So there are already existing standards. And then I thought, when I started this project, so there are already multiple standards, then do I need to add another standards on the top of those 20 standards existing already? And the answer is that this was really great starting point for us, but there are some gaps in those existing standards. So yes, we needed to kind of build on existing data standards, but then create another one. So we identified the gaps, and those gaps include, for example, like qPCR detection, amplified single species detection method. Those were not really the scope of any of these existing standards, but a lot of eDNA, it's not only about metabarcoding, but we also use a single species detection. So we worked a lot on that. And some sort of quality controls. Yamakawa-san identified that eDNA, the quality control is really important. Contamination is a big risk. So we wanted to also focus on that kind of part. So to fill those gaps, we developed these FAIRY guidelines. And so I'm going to walk you through what's in the FAIRY guidelines. To develop these guidelines, we put two main focuses. One is that we want to make sure the data, shared data, would have a complete information. So it's not going to be half empty like how it's been. And then we want to make sure the machine readability. So it's going to be interoperable. We're talking about not just 5 data sets. We want to manually format and then put them together. We're talking about a lot bigger number. So we cannot do it by hand. So it needs to be machine readable. We can put them all together and then reuse it. One section is about data components and the format. So we defined what components are needed. It's not only about the raw FASTQ files or OTU table. We need a metadata, we need a taxonomic table, and then multiple steps. So it's defined there. We want to make sure that format is all same. It can fit to multiple different types of study. Then one of the major part about this FAIRY guideline is this checklist. So it's basically a list of terms on the second column, term name. So that's the list of terms that people should, we recommend people to use in the metadata to describe how the samples were collected, how they were processed, how data were cleaned. And this checklist is quite big now because we are focusing on the complete information. Now we have more than 300 terms, and those were built on existing standards, as I mentioned. So half of the terms are from like Mixes, Darwin Code, those existing standards. But then the other half, we created new, it was new FAIRY terms because we identify gaps. When I say this 300 terms, I know everyone is thinking that this is too much and I don't want to do it, I can't do it. But I want to say, no, please don't run away from me. And because there are a few points that I want to mention here. Not all the terms are relevant for you. If you are doing a metabarcoding, then a whole qPCR section, those terms would be omitted from your template, and vice versa. If you're using a water samples, then the sediment core sampling methods will be omitted. So at the end, for your sample study design, you might have maybe 100 terms. And that's already still a lot, I know. But maybe a third of them will be just a unit. So you can see there is a sample size on the second row and a sample size unit. So Owashi-san and the student, they mentioned about this, the unit can be different in the metadata, then it's not going to be comparable. But we want to make sure like numeric variables are just numbers. And then you can talk about the unit in a separate column using control vocabulary. So in that way, it will be machine readable. So we really focus on those nitty-gritty bits, and that's why we ended up with a large number of terms. But if you start using that, and then I hope you would agree that it's actually not too bad. And then many of the information you fill can be reused across multiple projects that you are running within your lab or just for yourself. Right, so after we made this checklist and then defined the format and data components, we made this FAIRY practice step from 1 to 6 try to make it as simple as possible for users. So the first thing is to generate a template. And then you can use the templates from the beginning of the study, ideally, so that you don't need to, at the end of the study, you don't need to reformat everything. Then you can verify the data. There are multiple tools along the way. I'm going to talk a bit later. And then you can map the data. So the FAIRY format can be slightly different to database required format for reasons. So you can map the data to meet the database specific requirements, publish the data, and when you publish the data, you receive a DOI for your data. Then you can just put the DOI in your scientific paper data statement. Where to publish data is another whole component on these FAIRY guidelines. And as you mentioned, as we heard in the other talks, there are multiple very established international databases. So we basically recommend everyone to use these databases. They're doing a great job to implement eDNA dataset into theirs. But unfortunately, there's no one single database that we can just put everything like raw data and OTU table at this moment. So that's why some of the data, raw data, can go to INSDC, but then OTU tables and other stuff can go to GBIF and OBIS. So this is the description of that. I mentioned that there are tools to help people's life quicker, easier, and then also more accurate. These are examples. So we have our function at the top, FAIRY template generator, FAIRYator. So depending on your sample type or what assay you use, like is it metabarcoding approach or target species approach, how many assays you used, you will receive a template fit for your purpose. And then once you fill the data, then you can use the verifier. That's the second one, verifier. And it's a Python script that there's a web interface. You can drag the data in, and then it will analyze and find any errors and warnings, stuff like that. So sometimes this is a mandatory field, but it's missing, or this date format is not the ISO standard, and things like that. Then you can correct all the data until you don't get any more error message in that tool. Then mapping is, so we've been working with GBIF and OBIS group from the beginning of the project, but the formats are slightly different for a few specific reasons. So we created a tool called FAIRY to MDT. MDT is a tool to publish the metabarcoding data to GBIF. So you can use that to reformat and submit to GBIF and OBIS. What's exciting about this project is so there is now community uptakes of these FAIRY guidelines, and those communities are now using FAIRY templates, FAIRY formats, checklists, and then they are further generating, like developing more tools for themselves. And I said for themselves, but then it's for sure it's going to be useful for anyone doing eDNA. So the one on top, FAIRY sheet, it's a really cool one. So this top FAIRY generator, template generator, it gives you Excel sheet. But then NOAA team, they realized they want it on the Google sheet, because then field technician can fill the field component, then lab technician can fill the lab component, and the bioinformatician can fill the next one. So everyone can work on one platform. So they generated that, and then they published the data to NCBI, so they created the FAIRY to NCBI. Similarly, Mindaru Foundation, they published the data to ENA, so there's a FAIRY to ENA. All these scripts are now available. So I'm really excited that everyone is really taking what we worked on and then further developing more tools. And I realize all these tools are now a lot available, but scattered around. Some are RX scripts, some are Python scripts. So we now started a coding working group for FAIRY to unify these scripts under R package and a Python package. So I mentioned there are some community uptakes, and I'm really excited about it. So those are the examples of them. And then I spoke with ANEMONE group members, and then they're also implementing the FAIRY format, so I'm really excited about it. And this project is also endorsed by Aubon under UN Ocean Decade. And so we are working with the Aubon communities and making sure that they can also follow the FAIRY format. Then, okay, I mentioned about the journal team, so this is what it is. So we think that implementing this kind of guidelines through journal publication procedure is really one of the effective ways to change the scientist practice. So the great example is that journals implemented open data policy as a mandatory thing. And now open data is a completely normal practice for us. And they recognize that we also need to move from open to fair. So but then for authors, they say, okay, the journals are not recommending a fair practice, but how are we actually doing? How can we do it? Where is the guideline? And then domain-specific guidelines are required because each data set is very different depending on the field. So they accepted our FAIRY guidelines as a eDNA specific FAIR guidelines. And now we're doing the similar thing with the MBMG editors as well. And the other activities that we're doing is we're working with the data standards. So we've been working with them from the beginning of our project, but now we're really working closely. Every 2 weeks we're meeting with them and then making this FAIRY checklist under the Darwin Core and the Mixes schema. And why it's important? It's because these databases that I mentioned, they use Mixes and Darwin Core. So this kind of framework is going to really build a bridge and then connect all of us together. Multiple challenges are there, of course. Data quality. So once we have all this data from so many people, format can be standardized, but then is the quality really good? People use all sort of bioinformatic thresholds and things like that. So that's one thing. And I mentioned about this project with journals. We're trying to, highly recommend at this moment, but then if things go well, and then we want to mandate it. But is that really effective? How do eDNA researchers actually feel about it? I would love to bring that into discussion at some point. Then data sovereignty is another thing. Can we really openly share everything? How would the local people feel about the data sovereignty? Right, so last slide. Take-home message. These FAIRY guidelines are up and running, and then quite a few eDNA communities are using it now and integrating into their workflows. Lots of reusing opportunities are out there now, I think. Now we kind of established this FAIR foundation, and then more and more data are becoming FAIR, so it would be nice to actually reuse them. And stay tuned, as I mentioned, this website is the place to go if you want to look at what kind of updates and the more recent activities happening. We are still working really hard to continue improving. And then please reach out. This is really the community initiative, and we want to make sure these guidelines would fit, for many people's needs. So I would love to hear any feedback and then collaborate together. Thank you very much.